Great, thanks, Katie. So I'm excited to share with you all some work very related to the talk that Robbie just gave, thinking about the relationship between effect size estimates that come from multi-lab replications relative to those that come from meta-analyses. The work that I'm going to talk about today is the result of a work from a large team of people led by Christina Bergman and Maya Mather on the sort of statistics side of things. So imagine that you are a psychologist and you want to try to estimate the size of some effect, either because it's important for your theory or because it has some important sort of practical application. An important question to ask then is what's just the best way to do that? And if you think of sort of the classical sort of hierarchy of scientific evidence, you have sort of at the bottom, observational studies. Towards the top, you have randomized control trials. And at the very top of this pyramid, you have meta-analyses, which are, of course, sort of the statistical aggregation of effects from some existing literature studying a phenomenon. Another sort of approach to estimating an effect size is an approach we just heard a lot about, multi-lab replications, which don't exist on this pyramid, but surely exist sort of pretty high on this hierarchy. And these are coordinated replications across many labs that try to estimate the size of an effect, but do so by collecting new data. Okay, these different methods, multi-lab replications on the one hand and meta-analyses on the other, sort of have different strengths and weaknesses in terms of their evidential value. So on the one hand, meta-analyses are really great because they, at least relative to multi-lab replications, require relatively few resources. They have a lot of variability in population, typically stimuli and method, which is a plus in the sense that it allows you to explore generalizability of your effect, but can be seen as a limitation in that it sort of introduces a lot of variability in your effect size estimate. However, meta-analyses typically, the studies within them typically are not pre-registered. And the studies are likely subject to some amount of publication bias. In contrast, multi-lab replications are sort of way more resource intensive, both in terms of like the collaboration costs and in terms of just the fact that you need to collect a lot more data. They, unlike meta-analyses, typically have standardization of the stimuli and the method. And then they usually have some variability in population just because these studies are carried out in lots of different labs in different locations. And these multi-lab replications typically are pre-registered. So an important question to ask then is what's the relationship between these two aggregate estimates of effect size that come from these two different methods, multi-lab replications on the one hand and meta-analysis on the other. Naively, you might expect that these two effect size estimates should be the same. So if you're estimating the same effect for the same phenomenon using two different methods, you might expect that they should be roughly the same. Empirically, however, they're not. So there's some recent work by Kavarvan et al. From last year, suggesting that effect sizes that come from these two different sources are discrepant from each other. And in particular, that meta-analytic effect sizes tend to be a lot larger than multi-lab replications. 3 times larger was the estimate from this particular paper. One explanation or potential explanation for this discrepancy is the fact that meta-analyses, recall, are probably, the studies within them are subject to some amount of publication bias, whereas the individual estimates within a multi-lab replication are not. And so this could be one potential source of an inflated estimate that we get from a meta-analysis. However, some reanalysis of this data and some additional analyses suggests that while there may be some publication bias in meta-analyses, this can't fully account for this discrepancy that we see between effect size estimates derived from these two methods. So the question then is this why? We're left with this puzzle of why is there this discrepancy where we wouldn't really expect one between effect size estimates from these two methods. One intriguing possibility is that the discrepancy is due to the fact that there's just a lot more heterogeneity in these studies within a meta-analysis relative to a multi-lab replication. So, and these studies vary from each other along a lot of dimensions. So if you think of any given study, there are sort of many different dimensions that could arguably affect the size of an effect size. So, the method used, the particular sample, the stimuli, the analysis, the experimenter, the time of day, et cetera, et cetera. All of these factors could potentially influence how big your effect is, and this is likely a lot more variable in the case of meta-analytic studies. Not only are these dimensions more variable in the case of meta-analytic studies, there's reason to think that this variability could actually increase the effect size estimates for these studies, because these dimensions in the case of the studies within a meta-analysis may be adapted to the local context in which a study is carried out, where the local context may be the particular research question that a researcher is asking. It could be characteristics of the population, like the age of participants, the culture, et cetera. So by adapting features of the study to the local context, that could inflate the effect size estimates and potentially account for why meta-analytic effect sizes are larger. And so perhaps accounting for these moderators, this variability in the meta-analytic data, we might be able to better understand the source of the discrepancy between meta-analytic and multi-lab replication effect sizes. Okay, so with this motivation, we tried to understand this discrepancy in a particular case study that's important for language development, or at least thought to be important for language development, called infant-directed speech preference. And the question here is, do babies, very young babies, prefer to listen to infant-directed speech, what I'll call IDS, compared to adult-directed speech, or ADS? So adult-directed speech is sort of roughly what I'm using to talk to you right now, but infant-directed speech has these sort of different qualitative and quantitative features, where this has a sort of sing-songy quality. It has shorter utterances, higher varied pitch, longer pauses, and you've heard this if you've heard sort of any caregiver talk to their young baby. It has a sort of very different quality to it. And the claim is that infants prefer to listen to infant-directed speech that's designed for them, and that this special infant-directed speech may have sort of properties that facilitate language acquisition. Okay, so how do you study this preference in the lab? So the way it's typically studied is a baby is placed on their caregiver's lap, and they look at some interesting stimulus, like a checkerboard pattern. And you play for the infant adult-directed speech and infant-directed speech. And you ask, do they look at the checkerboard pattern longer when they hear infant-directed speech relative to adult-directed speech? And a longer looking time in the infant-directed speech case is taken as evidence that they prefer infant-directed speech to adult-directed speech. And the canonical finding is this, that infants indeed look longer. So I'm plotting along the y-axis, mean looking time to the checkerboard in the infant-directed speech condition relative to the adult-directed speech condition. We can quantify this preference that infants seem to have in any given study as an effect size, just as the standardized difference between looking time in these two conditions, infant-directed speech minus infant-directed looking time minus adult-directed looking time, standardized. Okay, so there exists a meta-analysis of this preference across the literature that was done in 2012. It included 34 studies across a roughly 30-year period looking at this phenomenon. And within this meta-analysis, they estimated the aggregate effect size to be about 0.67. And they also found, which is a pretty big effect size in developmental psychology, they also found that the effect varied as a function of a number of moderators that you might expect. In particular, the type of infant-directed speech that kids heard. So was it naturalistic, parents actually talking to their kids, to an infant, or was it sort of more artificial? The effect is smaller, as you might expect, for more artificial speech. It also varies as a function of age. So kids prefer infant-directed speech more as they get older. There's also been a multi-lab replication of this IDS preference called Mini Babies, which recently came out, in which each lab of many labs conducted their own replication that was sort of loosely based on Cooper and Asla in 1990. The particular design parameters were decided based on a consensus of a large number of researchers who actually carried out these replications. In total, there were 67 labs that participated in this effort and over 2,000 babies. So just to give you a sense of the breadth of this effort, here's a representation of the geographical distribution of all of the labs that contributed to this effort. In this particular multi-lab replication, the stimuli were constant across labs, and the DV was constant across labs. But there was some variation in the particular method that a particular lab implemented to test this effect. And in this multi-lab replication, they estimated the same effect, this infant-directed speech preference, to be 0.35, which is about half of what was estimated from the multi-lab replication. So just to represent this idea visually, here are the two effect size estimates from the two sources, the meta-analysis about 0.7 in blue, and the multi-lab replication estimate about 0.35 in red. So like previous work, in this particular case, the meta-analytic effect size is much larger than the multi-lab. And the question is why? Why is this the case? And so our hope was that we could get some leverage on this issue by taking this case study and systematically comparing effect sizes from the two sources, multi-lab replication and meta-analyses, for possible differences due to heterogeneity. And we were going to do that by coding the same set of moderator variables in each data source. So in particular, we coded 8 different variables for the two data sources. We coded the age of participants, the test language, whether it was native or non-native, speech for a particular participant, the particular method that was used, the speech type, infant-directed speech, simulated, synthesized, speech source, visual stimulus, was it related or unrelated to the speaker? The DV type, was it looking time or some other sort of related measure? And was it the target research question that was asked in the case of the meta-analysis in that particular paper? To analyze this data, what we did was fit a meta-analytic model, or fit a meta-analytic model that included both the meta-analytic data and the multi-lab replication data in a single model. And we fit 2 models. The first I'll call the naive model, where there was a single predictor, a single moderator, and that moderator just indicated whether an effect size came from the meta-analysis or the multi-lab replication. The second model we fit, I'll call the moderated model, included source as a predictor, but it also included the 8 other moderators that we coded that we think for theoretical or methodological reasons might affect the size of the effect size estimate. And this was an additive model. We didn't look at any reactions because of power limitations. The continuous moderator was centered, in this case just age, and the reference levels for factors were defined by the most frequent level in the meta-analysis. The model, unfortunately, was only able to converge with 3 of the 8 models that we looked at, specifically age, test language, and method. So the results that I'm going to be presenting for you from this sort of very initial analysis only includes those three moderators. And all of our analyses were pre-registered on OSF. Okay, so first, the results of the naive model. Here, just to start, is the distribution of effect sizes from these two sources, with multi-lab replications in yellow and meta-analytic effect sizes in gray. And the thing to notice here is, as expected, there's just a lot more variability in effect sizes from the meta-analytic data. Okay, so what do the effect size estimates, the aggregate effect size estimates, look like when we sort of reanalyze these two data sources? Here I'm just showing you that plot I showed you before, which reflect the original aggregate estimates from the published reports for these two data sources. And here are our estimates for meta-analysis from this model and the estimate from the multi-lab replication. So we replicate almost identically the effect size estimates from the published reports, which is good. So next here is but notably we still see the same discrepancy that we originally saw with about 0.32, and there's just a lot of variability, residual variability. So tau, which is the standard deviation of between study variability, or between standard deviation of effect sizes across studies, is about 0.35, which is pretty big, suggesting there's just a lot of unaccounted for variance here. Okay, so here now are the results from the moderated model. And I've now added a third estimate for each source, which is the square, that shows the effect size estimate after you account for the 3 moderators that we included. And as you can see, to our surprise, we still see a sort of sizable discrepancy between the effect size estimates from these two sources. And if anything, the difference seems to be bigger. Here are the model parameters from this moderated model. You can see that we sort of see an age effect. So as others have found, the effect seems to get bigger with age. And the other two, and you can see the parameters for the other two moderators there. So here, the sort of take home is we still have this discrepancy, even if accounting for some moderator variables. The discrepancy is bigger than when you don't account for these moderators, and the amount of unexplained variance is essentially unchanged. So one question you might have is perhaps this difference may be due to publication bias, and we can directly ask that question with these data. And the answer to that is it's probably not due to publication bias. So we did a couple of analyses that suggest this. So one is we calculated a corrected estimate for the meta-analysis, correcting for publication bias, and that estimate is actually a little bit larger, which doesn't reduce this discrepancy at all. We also did a sensitivity analysis using a method developed by Maya Mather and Tyler Vanderweel. Maya's going to talk more about this, her method, a little bit later in her lightning talk, but briefly, the idea is that you sort of assume a worst case scenario for publication bias, where statistically significant positive results are sort of infinitely more likely to be published than non-significant or negative results. And the way you implement this is you only meta-analyze non-significant or negative studies. And if you do this, the result is that it has to be the case that significant studies would have to be 8 times more likely to be published than non-significant or negative studies in this IDS meta-analysis in order to completely eliminate the discrepancy between the MA effect size and the MLR effect size. It's hard to know how to think about this estimate of 8 times, but it's much larger than estimates from other meta-analyses and also other meta-analyses within language acquisition in particular. All right, so just to wrap up, what we find in this sort of initial pre-registered analysis of these data, that even when analyzed within the same model and controlling for the same moderators, the MA effect size is more than twice as big as the multi-lab replication effect size. There's reason to think that this difference is probably not due, at least entirely, to publication bias in the meta-analysis. And so next, we're thinking about ways to try to address the convergence issue, because recall we coded 8 different moderators, but we're only able to meta-analyze 3 of them. And so we're looking to about ways to increase the amount of data we have. So one way we're doing that is trying to update the meta-analysis with more recent papers since 2011. And the other way we're doing that is thinking about including the data, additional spin-off studies and existing studies that have gone on to extend the initial multi-lab replication mini babies one. And there are a number of these. So one, there's an effort to look at infant-directed speech in bilingual children, an effort to look at test-retest reliability, many babies in Africa, and an effort to look at how this is related to native language versus non-native language stimuli. We're also thinking about a number of additional exploratory analyses that can be done to better understand the source of its discrepancy. So there's a lot, recall, of residual heterogeneity that we're not able to explore because we're only looking at 3 moderators. So one approach might be to fit separate models for these 8 different moderators that we're interested in. Another intriguing possibility is that there's this interesting difference in inclusion criteria between mini babies, or the MLR, and the meta-analysis. So in the mini babies effort, there was a pretty liberal inclusion criteria, namely that infants just had to contribute data to 1 trial in order to be included in the study, which is pretty liberal relative to studies that were included in the meta-analysis. And there's reason to think that making that criteria a little bit more stringent might increase the effect size. So here's just a plot of the mini babies data, effect size estimates varying the inclusion criteria. So you can see that as the inclusion criteria gets more strict, the effect size estimate goes up. So that's another avenue to explore as one possible source of the discrepancy. There are likely others. If you have thoughts about additional exploratory analyses that we might be able to do with our data, we'd be very interested to hear them. So with that, I'll wrap up. Here are some links to a number of the relevant papers that I talked about today. Thanks.
Thanks, Molly. And if you want to, I can try to copy and paste these links into the chat, but her slides are also up on the OSF, I believe, and you should be able to get at them straight from there if that's a little bit easier. But yeah, we've got about 10 minutes left, for questions and comments. So either write them in the chat or feel free to unmute yourself and just ask.
Can I ask a question?
Sure. Yeah, go ahead.
Yeah, so thanks for this interesting presentation. It's very interesting to see what is causing this heterogeneity, but I think it will also be quite difficult to figure out what the actual cause of this is. But if I understand your analysis correctly, you meta-analyze the studies in the meta-analysis together with the data of the labs from the multi-lab replication study, right?
Yep, that's right.
Okay, and then you have these different moderators, also, for instance, the different dependent variables. So did you also vary this in the multi-lab replication study?
So the dependent variable was the same in all effects, in all replications within the multi-lab replication study, which is looking time. Okay,
okay. And so how do these results, the results of the multi-lab replication, how do they relate to the seminal paper, the one by Cooper?
It's a pretty, let's see. Oops. Let's see if I can go back to.
So I'm especially interested in how the effect size of this seminal study was, how it's different from the multi-lab study.
Yeah, that's a good question. I don't know if someone who is more involved in the many babies effort knows off the top of their head how the aggregate effect size from many babies compared to the original Cooper and Aslan study?
It's hard to compare because the Cooper and Aslan method was very simple, but it was with newborns and 1-month-olds. So as a totally disjoint developmental population, and the stimuli were kind of really extremely exaggerated, about twice as exaggerated compared to ours. So they got a big effect, but it's very, very tough to do a direct comparison.
Okay, thanks. So then it looks like also the meta-analysis will be very heterogeneous because of this, right?
Right, which is, which I guess we think is not a sort of phenomenon that's particular to this IDS thing. We think is maybe a more general phenomenon and sort of the motivation for trying to code these moderated variables.
Yeah, okay, thank you.
Sure.
And it looks like you've got a comment in the chat. Can you see that? Would you like me to read it out?
Yeah, I see it. So the question is, or the comment is, I'm confused why you're so quick to rule out publication bias. I think significant results being 8 times more likely to be published than non-significant results is probably a low estimate. I think that's a very reasonable comment. I think it's sort of hard to know a priori what is a reasonable estimate of the ratio of non-significant to significant studies being published. I don't have good intuitions about that. We did, so that was sort of one analysis we did to assess publication bias. We also did this other analysis where we tried to sort of correct for publication bias in the meta-analysis. And there, the sort of, even after you correct for publication bias in the meta-analysis, the effect size is actually larger. And that's probably because there are more non-affirmative studies in the meta-analysis relative to affirmative studies. And so that doesn't do sort of anything to solve this puzzle of this discrepancy. So yeah. Another question is, what happens if you meta-analyze only the significant results from the multi-site replication? That's a great suggestion, and we haven't done that. But yeah, that would be a nice parallel analysis to do here. Thank you.
I have a quick question maybe about the Mini Babies project. It's super impressive to see how many labs and groups were involved. And I was just curious about your approach or your process in trying to find and identifying as many groups that were willing to do this. Were you involved in that process?
So I was not involved in that process, but there are a number of people here that were, that might, so maybe Martin or Mike or Christina, would you be willing to speak to that?
You can always go to the, I see, I think I saw Christina put a link in the chat, so I can always go to that afterwards. And I know that BITSS was actually involved in a couple of small ways of helping with the outreach. I was just curious of the kind of overall strategy, but I can follow up later.
I'm just gonna quickly say, because Mike signaled everyone was just being very polite. I mean, yeah, big credit goes to Mike actually, and also to Christa Bayers-Heinlein, the lead of Many Babies Bilingual, because there is already a big network there, and we have mailing lists, etc. And we just, especially Mike, talked about the effort at many conferences, and people just got very excited about it. So I just I think it's the community plus really engaged people who came together to make this happen. And there are many more Many Babies projects ongoing and coming up. So check the website. I'm going to put it in the chat.
Great, thank you.